Okay, hello, my name is Lilith and I'm one of your IAs for BIPN 160, and this is my review video for lecture 8. So my notes for this lecture ended up being pretty dense, so I will do my best to go over as much detail as I can. So in this lecture, we started to talk about the visual system. One thing that you want to make sure that you remember, because this might get you some easy points on the exam, is the anatomy of the cortex. So in this case, the primary visual cortex is right here on the occipital lobe in the back. You also might want to review the primary somatosensory cortex, which is right here. Remember that there is an S1, which is the purple. There's also an S2 somewhere down here. Remember the somatosensory association cortex. Know the Wernicke's area. Remember that Broca's area is on the frontal cortex over here. And also the auditory cortex is somewhere over here on the temporal lobe. So hopefully, if you're asked this, this will be easy points that you can get very fast. So talking about the visual system, the primary visual cortex is right here. This is also denoted as V1. And there are two streams of information that come from this cortex. The dorsal stream, which I've drawn here with an arrow. The dorsal stream of information tells us where objects are. So if I'm seeing an individual in front of me, I can tell by using the dorsal stream where this person is. And then the ventral stream, ventral means in the front, and remember anatomically this side of the brain is ventral and this side is dorsal. So the ventral stream tells us what we are looking at. So if I'm looking at an object in front of me, I can tell if it's a cup or, I don't know, a plate. Okay? Now, the organ that we use to be able to visualize things and see things is the eye, right? So make sure to go over and at least be familiar with these key anatomical parts that Dr. Root provided you with. So the pupil is, let me make that a little smaller. The pupil is the area of the eye right here in the middle. The pupil allows for light to come into the eye. The iris is the iris here, in this case blue, surrounds the pupil. And the iris is also a muscle that is able to contract and relax to dilate or constrict the pupil. The lens allows for us to focus light on the specific parts of the retina, so we're able to see. The vitreous chamber or area, which is right here. This is filled with aqueous gel, which allows for bending the light a little bit, but also keeping the form of the eye. The optic nerve comes out of the back of the eye right here, and we'll talk more about what makes up the optic nerve in just a few minutes. The optic disc is the area on the retina where the optic nerve exits the eyeball, and we'll talk more about this just in a minute also. The retina is the layer of the eye, this orangey yellow layer that is photosensitive, meaning it is going to sense the photons of light that are coming in through the pupil. And lastly, fovea is the part of the eye which is right here in this picture, and fovea is an area that contains high density of cones, and cones are a type of photoreceptor. So before I get to talking about those details, I want to make sure that everybody understands where we are located anatomically. So let's take this eye and flip it to looking at it on its side. So right here, we're looking at the eye from a side view. And in this particular picture, you see how this layer inside of the eyeball has been peeled off for us to visualize a couple things. So there are different layers. There's a sclera, a choroid, and you don't need to know those. What you need to know is that there is a retinal layer, and the retina is this orangey brown one. As you can see, the retina is the layer that is the outermost layer, meaning it is actually touching the vitreous gel. Okay, so it's this layer right here. And why do I want to make sure that you know that is because if we take a cut of the retinal layer and we expand it or zoom into it, what we can see is different types of cells in the retinal layer. Okay, so I hope you realized that this picture is telling us the type of cells that are located if we take a cross-section of the retina. Okay. Now let's look at the type of cells that the retina contains. So let's look at the right side of this picture. So this is the full size of the retina, and there are different photoreceptor cells in it. Okay, starting from the outermost layer, meaning the layer that is touching the aqueous gel right here, we have the ganglion cells. Okay, so these are the ganglion cells, and they are the closest to the gel, so that they're closest to the inside of the eye. That's very important to understand. Okay, then going inward, we have these bipolar cells. Okay, then going even more inward, we have the photoreceptor cells. There are two types of photoreceptor cells, the cones and the rods. Okay, the rods are responsible for allowing us to see dim light, or allowing us to see light when the source of light is very dim. And the cones allow us to see light that is very bright, or allows us to see color. Okay, so those are the distinctions between rods and cones. Now, there are other types of cells in here, but you don't necessarily need to know what they do. Just know that they exist here. So this horizontal looking cell is called a horizontal cell. Very easy to remember. And then we also have amacrine cells that are more down here. And they just help with modulating the retina and the signals that are going to happen in the retina. Okay? Now let's go back to this picture because I want to make sure that you understand exactly how light is able to turn into a neuronal signal. So if there is a light over here in front of the pupil, this light, or the photons of light, is going to enter the pupil. It is going to bend in some sort of way because it diffracts through the lens, and then it is coming over here and hitting the back of our eye, hitting the layer of the retina, okay, specifically at the fovea. And I'll go over what the fovea is again in a minute. So the photon is coming in and hitting the outermost layer of the retina. And if you remember earlier, I mentioned that the outermost layer is these ganglion cells. So the photon, let me take yellow, and also, so the photon is in yellow. It's coming here, it's coming here, it goes past through the ganglion cells, it travels back, it goes past the bipolar cells, and then it finally reaches the photoreceptors, either the cones or the rods. Okay, once it reaches the cones or the rods, it reaches the photoreceptor cells, it activates the photoreceptors, And once it is activated, I'll show you here in pink, the either cones or rods are able to now get activated and send neuronal signals down to the bipolar cells, which send neuronal signals down to the ganglion cells. And then once the ganglion cells receive that information, they allow for the information to travel down their axon. And you see how the ganglion cells and their axons travel down and then they curve over here. Okay. What happens to these axons of the ganglion cells? The axons of the ganglion cells end up going and collecting together to turn into the optic nerve, and the optic nerve later exits the back of the eye. Okay, so hopefully that made sense. So detection of light, or direction of light, so the light photon goes in this way, bypassing the ganglion and bipolar cells. It hits the photoreceptor cells, and then once the photoreceptor cells realize that a photon came in and activated them, the information, the neuronal information flows back this way, activating the bipolar and the ganglion cells. Okay, and once the information reaches the axons of the ganglion cells, these axons go together, and together they end up forming the optic nerve, which is cranial nerve 2. Okay. To show you exactly how cranial nerve is made, over here, the yellow layer of the retina contains all of the axons of the ganglion cells, and you see how all of them just come and collect right here from each side, and as they collect with one another, they turn into the optic nerve. So this is the optic nerve. Okay. Hopefully that made sense. Here on the left side, I've written some notes for you to remember. So the distribution of the photoreceptors on the retina is actually not equal everywhere. There is this spot on the retina that is called the fovea, and the fovea contains only cones. The reason fovea contains only cones is because the fovea is on a part of our eye, is on a part of the retina where the light coming in from the pupil can directly hit over here. Okay, so if we're looking at a bright source of light, then that light, if we're looking straight at it, then those photons are hitting exactly the area of the fovea. Okay, so at the fovea, we have only cones to detect bright light and color. However, the peripheral areas of the fovea, so everywhere else except the fovea, doesn't have as many cones and has way more rods. Why? Because the peripheral area doesn't get hit with as much bright light because the pupil is facing the fovea straight away. So everything on the periphery doesn't need to have cones, it needs to have a lot more rods. Okay? And the way you can test this is at night, if you go outside and you're in a pretty dark place and you look up at the sky, if you see a couple of stars in the sky, if you look straight at the star, sometimes the light of the star disappears, because if you're looking straight at the star, the light from the star is not strong enough to activate photoreceptors in your fovea. So you have to look at the star a little sideways, because if you look at it from sideways, the light from the star will actually hit the periphery of your retina, and your rods are better at seeing light that is more dim. So you have to look at the stars a little from the side to be able to see them. And the last thing that I want you to remember, or that you should remember, is the optic disc contains no photoreceptors. And why is that? Because it only contains the axons of the ganglion cells. Okay, the accumulation of the axons will later become the optic nerve and exit the eyeball. Okay. Now let's move on to this phenomenon called center surround. So center surround is a way for our brains to visualize or to understand whether we are looking at a dark spot in space or a lighter spot in space. Okay, so in this case, the green is light, and then the dark is no light. Okay, so how are we going to tell which areas are dark or light? We are going to be able to tell this by the phenomenon of having on-center bipolar cells and off-center bipolar cells. Okay, for you to understand this concept, there is a couple things that you should first remember. The on bipolar cells contain a receptor that is called M-glu receptor, M-glutamate receptor. Okay, and when this receptor is activated, when glutamate attaches to this receptor, this receptor allows for the bipolar cell to become inhibited. The off bipolar cells contain a receptor that is called AMPA receptor. And when glutamate attaches to the AMPA receptor, it actually activates the bipolar cell. Okay, so this is something that you should memorize for you to be able to understand what is happening in this image. The other thing you should memorize is this. When the lights are off, there will be a production of cyclic GMP in our photoreceptors. And if there is cyclic GMP, then the cell will be depolarized. And as you should know by now, if the cell is depolarized, it will release a neurotransmitter. Okay, now let's go over these two scenarios, starting with this one. So when there is no light, see it's dark over here, when there is darkness, so when lights are off, this cone becomes depolarized by using cyclic GMP. And when it is depolarized, it releases neurotransmitters. And in this case, the neurotransmitter is glutamate. So glutamate is released because the cell is depolarized because the lights are off. When glutamate is released, see right here, and I drew a couple more, a lot of it is released because this cell is depolarized. When there's a lot of glutamate, the on-center bipolar cells will become inhibited, right? Because remember over here, the on bipolar cells will become inhibited because their M-glu receptor will be linked to the neurotransmitter over here, and this whole cell will become inhibited. However, the off bipolar cell, when glutamate attaches to its AMPA receptor, this will be activated. And again, how do we know this? Because of what I asked you to memorize, right? When off bipolar cells get neurotransmitter, their receptor activates the cell. So in this case, the on-center bipolar cell will be inhibited and the off-center bipolar cell will be activated. If this is inhibited, it will not release any neurotransmitters. However, this is activated, so it will release neurotransmitters. And when it releases neurotransmitters, this ganglion cell is activated, and so the signal goes only in this ganglion cell. And this ganglion cell is also called the off-center ganglion cell. So when this signal goes into our brain knows that this signal is coming from an off-center ganglion cell. So our brain knows that this cone right here is not getting any light because it received the information from an off-center ganglion cell. Okay? When there is light, let's look at this scenario. When there is light, then this cone becomes hyperpolarized because lights are off, depolarized, but in this case lights are on. So this cone will be hyperpolarized. And when a cell is hyperpolarized, then it will not be producing as much neurotransmitter. And over here, I thought I erased some of the neurotransmitters, but let me do it again. So see, I'm erasing some of them because it's not releasing a lot of neurotransmitter. It's releasing like a few glutamates. Okay, now if there's not a lot of glutamates, right, not a lot, then there is no neurotransmitter, there's no glutamate to attach to the M-glu receptors or the AMPA receptors. Now, AMPA receptors of the off-center cell, the AMPA receptors, they need glutamate to become activated. And if there is no glutamate, this off-center cell does not become activated. However, the M-glu receptors right here, they need glutamate to become inhibited, but there is no glutamate. So this on-center bipolar cell does not become inhibited. And if something is not inhibited, then that means it is activated. When this is activated, it releases its own neurotransmitter, activating this on-center ganglion cell. When this on-center ganglion cell is activated, it sends neuronal signals to our brain. So our brain knows that this signal is coming from the on-center ganglion cell, and this tells our brain that the lights are on. Okay? Try to go over that by yourself to make sure that you can figure out what's being activated and inhibited. Now, if we look at the bigger visual pathway, meaning all of these photoreceptors are right here in the retina, and they send information down their ganglion cell axons, which later become the optic nerves, right? But what happens to this optic nerve after it leaves the eyeball? Well, what happens is that the axons that are coming from the edges or the periphery of the retina, these axons end up going back, getting to the optic chiasm, but not crossing, staying on the same side. Same over here. It stays on the same side. But the axons that are coming from the nasal side go down the optic nerve, get to the optic chiasm, and they cross. Same here. This one comes to the optic chiasm and it crosses over. Okay? Know that whatever you see in front of you is the visual field. So that means we have a left visual field and a right visual field. The left visual field in this case will relay light onto the nasal side of this eyeball and the temporal side of this eyeball. And I've color-coded them for you also. And the visual field of the right side relays light to the temporal side of this eye, and it relays light to the nasal side of this eye. Okay, so in case that was hard to understand, let's say I draw a heart over here. So if the heart is on the right side, the neurons or the photoreceptors that are going to be stimulated because we are looking at this heart on the right side, those photoreceptors will be on the temporal side of this eye and on the nasal side of this eye. Okay? Gonna erase that heart. Okay, so now that we see what the pathway is for the optic nerves, let's see where these pathways go to. Well, the first stop after the optic chiasm is the left geniculate nucleus. So the optic nerve leaves the eyeball, goes to the optic chiasm, some part of it crosses over, another part stays on the same side, and both sides go to the lateral geniculate nucleus of the thalamus. And the lateral geniculate nucleus is portrayed over here. The lateral geniculate nucleus is layered, and layers 3 to 6 are called parvocellular layers, and they respond to color and form. And layers 1 and 2 are called magnocellular layers, and they respond to motion and contrast. After these neurons have reached the lateral geniculate nucleus of the thalamus, they relay back to the occipital lobe where V1 is located. V1 was the visual cortex, the primary visual cortex. Okay? And one thing I forgot to mention, the neurons that project from the lateral geniculate nucleus to the V1 are called the optic radiation. I'll write it over here. Optic radiation. Okay. Now that the signal is in the V1, in the primary visual cortex, what happens in the primary visual cortex is that the cortex itself receives information from both eyes in an alternating way, meaning one patch of the visual cortex receives information from the left eye, the patch next to it from the right eye, then from the left eye, then from the right eye. Okay? So again, in the visual cortex, our brain is again sort of mapped out in terms of which eye is bringing in the information. Okay. Another thing that Dr. Root mentioned, he'd like you to know, is if there was a lesion to any of the parts of the optic pathway or the visual pathway. So if, for example, let me zoom over here, if there is a lesion that is caused on the left optic nerve, then you will not be able to see completely on from the left eye, but you will be able to see from the right eye. Okay, if the cut has happened or the lesion has happened at the optic chiasm, so nothing can cross to the contralateral side, what you will be able to see is only the nasal parts of your visual field. So it will be, I think it is called tunnel vision, right? If you look straight ahead, you will have a hard time seeing things that are far to the left and far to the right. You will only see what's right in front of you. If there is a lesion where letter D is right here, so this is the optic tract, if this is cut, you will only be able to see things that are in your right visual field. Okay, and I'll go ahead and explain this one in case that's hard to understand. So if the left side, if this tract is damaged, right here, if this is damaged, then how can we tell what is being affected? We have to track where the neurons are coming from. So if the neurons that are ending up over here are coming from this, right? And this, then if we cut right here, we will not be able to see anything that is happening over here, which is our right visual field. Okay, hopefully that explanation made sense. And then to be very honest with you, didn't quite understand this quarter visual field thing, but just remember that if one of this either top or bottom pathways of optic radiation is lesioned, then you will able to see the opposite. So I said that wrong. So if the lesion happens at the bottom optic radiation pathway, then you will not be able to see the top corner of your visual field. And if the lesion happened at the top, then you wouldn't be able to see at the bottom corner. Okay, so same idea for all of them. If this is a question that you're asked, just make sure to pull up a picture of this and just look to see where your visual field is, which one you're interested in terms of the lesion, and which pathway is being affected. This one, this one, this one, or this one. Okay. And then on this page, to finish off this lecture, I just wrote some information in purple boxes to help you just easily remember it. I don't think you need to know these experiments in detail, but just going over them and going over the main idea should probably be sufficient. So remember that the famous experiment by Hubel and Wiesel showed that the primary visual cortex, also denoted as V1, responds to lines of particular orientation, meaning our primary visual cortex is able to pick up information by seeing different orientations of objects. So if I draw a vertical line and a horizontal line, then different areas of our cortex will be able to respond to that, and we can tell exactly what the orientation of those lines are. Okay, and the research with this one was with the cats, where they implanted electrodes in the visual cortex of the cat, and they showed the cat different oriented lines, and they recorded the neural activity in the V1 cortex. Okay, the next area that you should remember is the MT area, or V5. Know that this responds to motion detection. And the experiment that was done for this one was with the monkey, where they showed the monkey different moving dots, and if the dots were moving, all the dots were moving together in one direction, then the monkey learned to let the researchers know somehow that I understand that they are moving in the same direction. And so that is happening in the V5 area. 3 things that you should know about this is some techniques that the researchers used. So first, of course, they recorded to see that the MT or V5 area actually responds to motion. Then what they did is they lesioned this area, and they realized that this actually causes impairment of motion perception. So they realized that this area is required for motion detection. And then they also stimulated this area, and when they stimulated that, they saw that the area is sufficient for motion detection. And this type of experimentation was taught to you in lecture 2 or 3, I think. If you want to see if something is required, you want to lesion it or disable that part of the brain, and if that function is no longer happening, so in this case motion detection was impaired, then you know that this area is required. But if you want to see if this area is sufficient or not, you want to activate that area. Okay. The area of STP responds to face recognition. Not much to know about that really, just they did a quick research where they showed people different fast-moving images and then every time you recognize the face, you click the button or something. So you don't need to know the details. Just know that STP has to do with face recognition, and if you lesion this area, then the individual is no longer able to recognize faces, even if that person who is they're trying to recognize is sitting right in front of them. They can't understand who that person is. And then finally, the area of V4 responds to perception of color, and that's, I don't think you need any more details than that. Okay, a little good luck from me to you. I'm glad that Dr. Root put the answers over here for you, so I won't go over them in the video since it'll just be me reading them, and I think I can use that time to make another video for you for lecture 9. So please go ahead and read these questions on your own and understand the answers, and if you have any questions, don't be shy to ask. And I've also cut and pasted the learning objectives for you on the bottom here. Use the learning objectives as a checklist for you, like be able to identify the anatomy of the eye. Do I know this? Great. Next. And do all of that to make sure that you're as ready as you can. I think that's it. If you have any questions, please email me. I am also studying for a few exams for Tuesday, so I'll try to email you to the best of my abilities before Tuesday. But I will go ahead and put lecture 9 soon, either later tonight or worst case scenario tomorrow morning. And I will possibly do another video talking about some important topics that you should pay special attention to. Okay, that's it from me. My name is Lilith. Let me know if you have any questions, and you can find the PDF of these notes in my IA page. Thank you.